In this video, we will discuss about protein synthesis and degradation in plant cell. So far in our previous videos are based on a mammalian cell mostly. However, in this video, we will cover the protein synthesis and degradation in the plant cell. It is mostly similar to the mammalian cells. However, in order to clear anyone's confusion, we will cover this thing here. So we will start with the pool of amino acid that are present in the cytosol. Consider this as a cytosol. And so pool of amino acids, first the translation of the mRNA occurs with the help of ribosome. So that this pool of amino acid will form a chain of amino acid following the sequence that is present in the mRNA. After that, the protein or during this translation, some of the proteins are destined for the to the endoplasmic reticulum, and this is determined by the signal. This is determined by the signal sequence that is available on the ribosome. So those proteins will move to the endoplasmic reticulum where folding and post-translation control occurs, and after that it will move to the Golgi apparatus for further maturation of this post-translation modification, and this modified protein will then transport it through these vesicles to their final location. That is either plasma membrane, or they are secreted outside the cells, or some of them will move into the organelle for which they are destined to, such as mitochondria, nucleus, etc. So this is the regular process. However, what happens is this during post-translation in the endoplasmic reticulum, when the proteins are not able to acquire their native structure, or even in the not even in the even after this translation of this protein, if they are not able to acquire their structure, then they will be degraded, and and again recycled to a pool of amino acid, and this process is usually done by ubiquitation or proteasomal degradation. This is the proteasome and this is the ubiquitin, which will target them, or which will target them to move towards the proteasome for proteasomal degradation. And some of the proteins may be targeted to the organelle before they reach the endoplasmic reticulum, or those proteins that are not destined to move to the endoplasmic reticulum for post-translation modification. Some of the proteins may directly go to the organelles, and translation of a small amount of proteins occurs in the mitochondria and in the chloroplast also. This is shown by this red arrow. All the red arrow shows the protein translation, and the black arrow shows the protein degradation. And and since and also since this is the black arrow from these organelles, that is chloroplast or the mitochondria. So some of the proteasomal protein degradation occurs in these organelles also, and they will again recycle back to the pool of amino acid. And if the organelle is damaged, if some organelle is damaged or is in excess, then they will be moved towards degradation, or they will be diverted towards degradation through this autophagy process. And all this will be controlled, and all this protein synthesis and degradation will be controlled by this mTOR, signaling pathway. Yes, it will determine whether there is a excess availability of proteins or organelle, or there is a there is a deficiency of some proteins or organelle. Based on this, the mTOR signaling pathway will get activated and try to balance the protein synthesis and degradation in the plant cell. So, and this thing is similar to the process that happens within the mammalian cells. That is translation of process proteins occurs in the cytosol, and if the signaling pathway signals them to move into the endoplasmic reticulum, they will go to the endoplasmic reticulum. Then the further post-translation modification or protein folding occurs, and then it will move to the Golgi. Then the mature protein will be destined to its final location with the help of vesicles. That is either they are destined to the secreted protein or to the transmembrane protein, or they might be destined to the different organelle of the cells. So this is how the protein synthesis and degradation occurs in the plant cell, and which is more or less similar to the protein synthesis or degradation that is occurred within the mammalian cells, almost like 90% similar. There could be difference in the post-translation modification occurs in the plant cell that we have discussed in the during or in the video in which we are discussing about bioinformatic bioinformatic platforms for the production of biotherapeutics proteins. You can go through that video to understand in depth about the difference in the post-translation modification compared to the mammalian cells, and why this plant cells is not a best option to develop the biotherapeutics. So I hope you have understood something about this, or basic about the protein synthesis and degradation in plant cell. If you need to understand this thing in details, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.